What makes a skin cell different from a brain cell? And can one type of cell be changed into another? As a biochemist and stem cell biologist, Professor Jose Polo is working to better understand these questions.
So that's why my lab works in so many aspects, but actually we are always working in the same thing, how cell A becomes cell B.
While every cell in the human body has the exact same genes, the epigenome controls what genes are expressed, which determines what the cell becomes. By harnessing these epigenetic processes, Professor Polo and his team are able to reprogram one cell into another cell type.
And we found a way to control how those genes are open or closed. We use these proteins that are called transcription factors that are like, I call it the librarians in that library that controls which books are open or closed, which genes are active or not.
Cellular reprogramming can use viruses or RNA to introduce these transcription factors into the cell. Professor Polo's lab has used these techniques to grow everything from lung cells to placental cells and even models of the early human embryo.
This is the pregnancy test of the cells?
Yes.
So it's already positive?
It's already positive.
During the pandemic, we were able to check if, for example, if the SARS-CoV-2 virus, the virus that caused COVID-19, was able to infect placenta cells. But we didn't need to collect placenta cells. We were able to make the placenta cells directly.
In 2012, Sir John Gurdon and Professor Shinya Yamanaka received a Nobel Prize for their foundational work on induced pluripotent stem cells, or iPS cells, which have transformed the field of regenerative medicine.
Induced pluripotent stem cells, or iPS cells, are super important because they are able to differentiate into almost every cell type of the body, which it means that you can take skin, reprogram them into iPS cells, and then from those iPS cells, we can convert them into heart cells, brain cells, blood cells, etc., etc.
But as a postdoc researcher, Professor Polo found that those cells carried an epigenetic memory of what they used to be, a discovery that was simultaneously being made by another postdoc researcher at the time, Academy Fellow Professor Ryan Lister. Years later, when they both came to Australia, they worked together, firstly to figure out why that memory was retained, and then to develop a way to wipe it.
And with that, we created a method that gives rise to corrected iPS cells, which we show that they differentiate more homogeneously into all the cell types. They don't carry these epigenetic aberrations, and in general, they are easier to work with.
It's research with significant implications for disease modelling and drug screening.
And you can apply this to everything: cardiac problems, muscle problems, any problem where the problem is a cell that is not having the function correct. You could model it in vitro now, directly from a patient, and then test drugs.
Groundbreaking work that has now seen Professor Polo elected as a Fellow of the Australian Academy of Science.